that brief lesson review that you've given us, Edwin. It was really good, and it is impressive that you guys, you follow, and you really know what you're doing, okay? And it's worth it. So since when we introduced our chemicals of life, we have really trekked, where we started with inorganic ions, where we talked about water, mineral salts, and stuffs like that. And then we talked about organic compounds, where we were able to talk about carbohydrates and all the types, the monosaccharides, polysaccharides, stuff like that. And then when we finished carbohydrates, we talked about lipids. We were able to talk about the structure, the amino acids, the fatty acids, the types of fatty acids, the essential, non-essential, the puffers, the muffers, the saturated ones. We saw how the fatty acids condense together with the glycerol molecule to form a fat. Basically, all that. I hope you know all that. And if I give you any question as far as chemicals of life is concerned, then you must be able to do. And like I told you, I've posted enough questions in the classroom, in the Google Classroom. Okay? Always go there and do. Always go there and do. And if, like always, if you can contact your friend, your friend might be there and the friend needs help, you can let them know. Okay? You can let them know and join our Google Classroom, and we make life simple for them. So now we are here on team proteins. Okay? We are on proteins. And I was amazed with these models, where this is a space feeding model of polymerase enzyme, and this is a space feeding model of a transmembrane protein. And I got in these ones from BS 5th edition, page 980. They were really amazing. Okay? They were really amazing. And so you can have a look. So this is our DNA polymerase, and you can see this is the DNA. So this is the basically, anyway, we shall talk about DNA synthesis. Okay? We shall talk about DNA or protein synthesis and DNA replication, stuff like that, where we shall be able to talk about polymerase. So now imagine your life without polymerase. Imagine your life without these transmembrane proteins. Okay? These transmembrane proteins. Imagine how life would be. So in simple terms, imagine your life without proteins. Okay? So this is another class of organic nutrients that really do perform some important function, or actually functions, not just function, in our bodies. Okay? They are so important in our bodies. And by the end of today's lesson, but by the end of the lesson proteins or protein lessons, we shall really appreciate how important these proteins are in our bodies. Okay? Yes. So as usual, feel free to drop your questions in the chat box, and I'm always happy to attend to your questions, guys. So proteins, the organic compounds of very large molecular mass. Large compounds formed by condensation of many small units called amino acids. We shall see how condensation occurs. And they are insoluble in water, but rather they partially dissolve to form that colloid solution. For instance, you try to dissolve an egg. Try to dissolve an egg in water. Okay? And see what may happen. The egg will dissolve to form a given solution. Okay? But the solution, it's not like other solutions. Like when you dissolve copper to sulfate in water, when you dissolve salt, sodium chloride in water, like when you dissolve sugar, sucrose in water, or glucose in water. The solution here is clear. It's a clear solution with a color depending on the cation in that salt. But now, in this case, it will partially dissolve, yes. It's not that it is completely dissolved, that you can filter it out, that you can decant water off, or that it will form a layer like when you dissolve oil in water. No, it's not that way. But when it is dissolved, it will partially dissolve to form that colloid solution. Okay? So that is a unique property of proteins, and all proteins behave in such a manner. Now, unlike fats and carbohydrates, okay, so for proteins, they do have carbon, they do have oxygen, they do have hydrogen. But in addition to these ones, they also have their nitrogen, they also have their sulfur, and sometimes phosphorus, depending on a given protein and a given amino acid. Okay? So these ones, they do behave differently from the rest, in a way that in addition to carbon, hydrogen, and oxygen, they also add their nitrogen, they add their sulfur, and they add their phosphorus. And we are going to see the structures of amino acids. They are the most abundant in nature. Okay? They are the most abundant in nature. E.g., you may have a single bacterium, for instance, might have around 800 different types of proteins. And man, you who is watching, who is listening to me right now, you may have around 10,000 different types of protein. So you can imagine. And all these different types of proteins, they are all derived from amino acids. Okay? So the diversity is due to these amino acids that are condensed together to form the proteins. Okay? These several amino acids that do condense to form a protein, they are the ones that bring about the diversity, okay, of these proteins. And the formation of any given protein, it should occur with care. In that, in the case of where amino acid A, just amino acid A, if where you're supposed to put amino acid A, you put there B, then everything becomes distorted. That now this protein will totally be different from this protein. So therefore, when you are forming these proteins, you must form them with care. Yes, somebody has a question?
Yes.
Anta, good morning.
Good morning.
What's the question?
What's the difference between amino acid A and amino acid B?
Oh, okay. It's okay. What is the difference between amino acid A and amino acid B? We are going to answer that when we reach amino acids. Okay? And you'll notice, you'll see the difference. And you'll be able to appreciate. Okay? Yes. But these amino acids, they are different. Actually, they are roughly 20 known amino acids. 20. 10 essential and 10 non-essential. So there are 20 known amino acids. And there's these 20 known amino acids that are used to form these 10,000 types of proteins in your body. Imagine 20, just 20, but they are arranged in different combinations to form those various types of proteins. And we shall really appreciate. This is some interesting topic that I usually enjoy when discussing it, when teaching it. It's really nice. It is really nice. And I hope we shall have fun together. And so, that's brilliant. So, these proteins are specific to each species, hence they determine the specificity in the characters, okay, in this species. That's why species A is different from species B. That's why species A is different from species B. For instance, let's talk about the specificity. Do you know that you cannot fertilize with a cow? You successfully have sex, you successfully copulate with a cow, you eject in your sperms. Do you know that you cannot successfully fertilize with a cow? Okay? You cannot. Studies have successfully put it forward that the ovum. Have I ever discussed this with you? No, I think it's Form 6s. I think it's Form 6s or no, it's you when you were studying cell biology. It is you. So the ovum, it has a layer that is impermeable to these sperms. It is a layer that is made up of fibrous tissues. They are just fibers. Okay? And this layer is called the zona pellucida. You have their glycoproteins, many of them. Now, this layer is impermeable to sperm cells. And it is actually this layer that brings about the specificity among different species. That if you copulate, maybe if a woman copulates with another man, same species, they are all Homo sapiens, they will fertilize and produce a zygote. But if a woman copulates with another animal, maybe which animal? Okay, maybe a dog, maybe a cow, then these two cannot fertilize. Why? Because the sperm cells of the dog cannot penetrate through this zona pellucida. And if they cannot penetrate, then there is no way how the nucleus of this dog's sperm is going to fuse with the nucleus of this woman ovum. And so you are considered two species. Now the question is, how do these organisms in the same species do it? They are proteins. Okay? That within the ovum, you have their protein receptors, you have their specific receptors. Okay, specific receptors, where the receptors on the sperm must come and bind to. Okay, that when the receptors in the sperm bind to the receptors in the ovum, then this will induce an acrosome reaction where you release enzymes. Yeah, I remember when I talked about this, when we were talking about the virtues, cell virtues, the phagocytic virtues, those ones, the different functions, when we were talking about lysosomes and lysosomes. That's when we talked about this. So you release these lytic enzymes, they digest this zona pellucida and the sperm penetrates. So that specificity is brought about by the proteins, and it's really good. It is really good. Otherwise, it will just be confusion. Actually, that's what I've heard. Not stored in organisms. Yes, we don't store. No organism stores proteins except in eggs and shells. Use it to form new tissues. For instance, if you consume too much meat on Christmas, what happens? Most of you people usually have a running stomach, usually have those complications. Why is it so? It is because you don't store proteins, so the too much meat that you've consumed, okay, must be deaminated and converted into urea, and then you excrete urea out of your body. Okay, as simple as that. You don't store proteins apart from in eggs, because why eggs? The eggs, that albumin, it is stored proteins for this growing embryo. Otherwise, if you don't have it, then the embryo cannot grow in that egg. Then form structure basis in all living cells. This is it. There's proteins. They are the ones that form structures, form cells, that cells make your body structures different from them. For instance, you can think of keratin, okay, which forms hooves, fingers, fingernails, not fingers, hair, and stuff like that. The scales and stuff like that. It's a protein, okay? So really, these proteins, they must be given enough attention, enough care, due to their diversity and due to their diverse function. The building blocks are amino acids, yes, and somebody have said, how are these amino acids different? And you are going to see. So just like you saw in carbohydrates with many glucose molecules condensing together to form the amino acid, just like how you saw fats, the fatty acids condensing together to form the fat. Sorry. Also here, these amino acids, many of them, they do condense together. Okay? They do condense together to form proteins. And you're going to see the processes. Okay? We are going to see. So from that brief overview, then we are free to talk about amino acids. And like I told you, me, I don't like complicating things. Just from the name, amino acids. You can read it. You you can really see, you can really write the structure even. Amino, from that basic chemistry that we've had, you know that this is the amino group. Okay? From our basic chemistry that we've had again, acids, you know we have one organic acid, okay, which is a carboxylic acid. Okay, so therefore if we combine these two, can't you really form the amino acid? You can. So you have the amino group, and there is no harm if I write this in this format, and then you have a carboxyl group somewhere. Okay, yes, then you have a carbon that is joining them. Okay, and this carbon, it is joined to the hydrogen here. I think this is the alpha hydrogen. Okay, and you have a certain group here. Let me represent it with R. Now this R is not an alkyl group. Stay warned, like what you use in organic chemistry. This R is not an alkyl group, but it is any group. Now, all amino acids have this, and they have this, and they have this, and they also have this carbon. All of them, all the 20. But what varies?
Teacher, all the what?
All the 20 amino acids. I say there are 20 known amino acids. Okay? Some books say 22. But what's confirmed? So far it's 20. So all the 20 amino acids, they have this group. So they have this, they have this, they have this, the way they are. But what varies? It is this group. And therefore, it's this group here that determines which amino acid is going to be which, and which amino acid is going to be which. For instance, in methionine, for instance, okay, in methionine, this other group is just CH3, okay, but in other amino acids, it can be as complex as H. Then you have the CH3, and then the SCH2, something, and then you combine this to this carbon. So it can be as complex as, depending on the given amino acid. So if you have understood all this, then you know what amino acids are. So briefly, they are organic molecules with both an amino group and a carboxyl group. This is the amino group. Here it is NH2. It comes from ammonia, but this is an amino group. Amino group. And then a carboxyl group. And this is carboxyl group. So from your basic understanding of organic chemistry, you can tell that this one is a carboxyl group. So basically all amino acids have those two. And actually it's because of those two that we have given them that name, amino acids. Then in addition to those two, you have the general formula of that, okay, where you have the amino acid, and now the amino group and the carboxyl group, they are joined by a carbon atom, okay, which is called the alpha carbon. Now it is that carbon atom that joins both the amino group and the carboxyl group. Now on the carbon, like how I illustrated, you have a group which varies from amino acid A to amino acid B to amino acid C to amino acid D up to the 20th amino acid. And so we can have their basic structure, I think. Yes, this is the structure. Whereby you have the hydrogen here. This is the amino group. And then this is the carboxyl group. That's why it is called an amino acid. Why acid? Because of this. This is an organic acid. And why amino? This is an amino. And this is the central carbon atom. Okay? The central carbon atom, which has the hydrogen, like how you can see, and then the group that varies among all the 20 amino acids. Not so?
Teacher,
Yes.
why does the carbon, like the carboxyl group, why is it separated and the other upper side, why does it have a double bond with oxygen?
Oh, that's good. They were trying to show you how it is bonded to form that group. Okay? Yes. How
So
bonding?
what is the importance of that double bond between the carbon and oxygen?
It is how carbon is bonded to the two oxygens. You remember the organic chemistry, the concept is carbon must have 4 bonds. Okay? So it has bonded one with this alpha carbon and bonded with another. This is OH, the hydroxyl group. Then it has a balance of how many? 2 bonds. 1, 2. So it has a balance of 2. So the balance is going to be bonded with oxygen here. And also oxygen forms 2 stable covalent bonds, if you remember. Because it has 6 electrons in the outermost shell. So it needs how many? It needs 2 to have that octet structure. So that's why oxygen forms 2 covalent bonds. Not so? So that's why a carboxyl group is illustrated as such. Is it so? It is okay, eh? Good. So those are your amino acids. Don't forget this because it is going to be key. It's really going to be key as far as protein is concerned, as far as protein synthesis is concerned. Because after now understanding proteins and how they are formed, you need to study how these various proteins are formed and how they can control certain issues, certain things. And why, if a mutation occurs, then a mutation is going to change the overall protein. So we are going to see all that. So this structure should really be part of it because it's key, okay, as far as protein, biology, protein stuff, protein synthesis, proteins, biochemistry is concerned. Now that amino acid is some unique structure, it has some unique features in that it can dissolve, I mean the amino acids, they are soluble in water, and when they dissolve in water, they do ionize to form ions. However, one unique thing is on the same amino acid, you have there the amino group, which is basic in nature, and you have there the carboxyl group, which is acidic in nature. Are you seeing that? You have the amino group. Let me write it. The amino group. And this group is basic in nature because it has a lone pair of electrons. And the acid, which is CH and then OH. So therefore this will behave as an acid and this one will behave as a base. So as a result, when you put your amino acid in acids, it will be able to live there, and also in neutral it will also be there, and in basic it will also be there. However, in different states, in different conditions, in different appearances. In that this amino acid or this protein in acidic medium, it will be in different format. But when you change it from acidic medium to basic medium, it will also change the format. And you put it in neutral medium, it also changes. So that implies if the amino acid is okay in an acidic medium, then let it stay there in an acidic medium. If it is okay in basic medium, then let it stay there. For instance, that's why change in pH, it alters the enzyme activity. And we are going to see factors. We are going to talk about enzymes and we see factors. So think
Teacher,
about in your mind how these enzymes, how these proteins are going to be affected by the pH.
teacher,
Yes.
when you're speaking about
About?
you're speaking about basicity of what the amino group, you said you talked
I said?
about lone pairs.
Yes.
You mean that you say that because it has lone pairs? Get it?
Yes.
I want you to explain to me why.
I say that it has lone pairs, eh?
Yeah, like give you another example of a
Yes,
basic compound,
it's
maybe
okay.
like sodium hydroxide.
Okay,
Then
sodium.
you
It's okay. Go ahead.
first explain. I just want to know why because it got me curious when you're speaking about the amino group.
It's okay. Now, basicity is the ease of accepting a proton. As simple as that. The ease of accepting a proton. And a proton is just now this is chemistry, but it's okay. It's better. It's good I teach chemistry and biology, so it's okay. We can switch at any time as long as it's good for the understanding of a given concept. So basicity is the ease of accepting a proton, and a proton is simply hydrogen that has lost its electron. Its electron, in fact, it's one. So this hydrogen does not have any electron, so it is a proton. So it always seek for electron-rich centers. Okay? And so, therefore, for you to be able to accept that proton, then you must have the lone pair of electrons. You must have electrons. And the ease at which you're going to donate those electrons, it determines your basicity. So the higher the ease, the higher the basicity. Okay? So now, the amino group, this amino group, it has lone pairs of electrons. You see, it's nitrogen that is bonded to three hydrogens. You have hydrogen here, you have hydrogen here, and then the other bond is with the carbon. So it's like this. See something. Now, nitrogen has 5 electrons in the outermost shell. If you want to know, we have the Vc, and we shall study this in physical chemistry. Okay? If you want to know how many lone pairs of electrons will an element remain with, for example, nitrogen, you're going to consider the electrons in the outermost shell. Now this is too much of chemistry, but it's okay. Then minus the bond pairs, and the bond pairs, it is 2, okay, times you've formed how many electrons. So this is 1, 2, 3. So there are 3 bonds, okay. So you can say 2 times 2, 2 times 3, okay. However, a covalent bond is formed by sharing, okay. It is formed by sharing. So we shall subtract the other 3 electrons that are formed by sharing. Not so? Yes. So in simple terms, it is the same as saying 5, which is the outermost, minus 1, 2, 3 bonds. So you have 5 minus the 3, and you remain with 2 electrons out of 5 that remain untampered with, that remain untouched. And these 2 electrons, they are the ones that contribute to the lone pair, okay. And so now they are the ones that are donated to the to the proton when it's coming to join this nitrogen. And so the ease at which you donate these electrons determines how high your basicity is going to be. Okay? So also in an amino acid, this one is going to be basic and this one is going to be acidic. So basing on how, okay, which solution you are, it will determine how these two are going to appear. Anyway, we still have a lot of concepts to grasp. Let's proceed. Okay? And so that's how it is going to appear. In that if this amino acid of ours is put in a basic or is put in a neutral solution, is dissolved in water, for instance, okay, then it doesn't stop this to lose its proton because it is an acid, and you know how acids behave. The acids behave by losing, or an acid is any solution that loses hydrogen ions as the only positively charged ions. So in an aqueous solution, this one will lose the hydrogen ion, okay, to the aqueous solution. Now this one being a basic group, this group being basic, then the nitrogen, it will receive that hydrogen ion that has been lost at this stage or from this end, okay, whereby it will donate its 2 lone pairs of electrons and forms NH3 with a positive charge on the nitrogen, because it's the nitrogen that has lost the electrons. Is that okay? Is that okay, really? Yes. And so when that happens, if the equilibrium is attained, okay, you reach a point where you've attained the equilibrium between these two. That is if maybe you are in a neutral solution, because when you are in a neutral solution, you can easily attain the equilibrium. But when you are in a basic, then you have more of OH. Then this one, the OH ions on this, they will be given, and you have more of this. When you are in an acidic solution, then the OH ion, the hydrogen ions, they will be absorbed by this, and you have more of this than how you have more of this, how you have this. But when you reach a given concentration where the positive charges on the amino group is equivalent to the negative charges on the carboxyl group, then the net charge is going to be 0. Okay? Net charge is going to be 0. So the overall protein is considered to be not charged. It has a charge of 0. And so such a compound, okay, that can form, it's one compound, but it has ability to form both positively and negatively charged ions. One compound, but can form both positive and negative charges. Then that compound is considered to be a zwitter ion. Okay? It's a zwitter ion. And so the overall charge is going to be 0. And this feature, my dear, is so important inside your body, for instance. It is so important inside your body. Maybe let me ask you a bit. Try to identify how this feature is important in your body. Try. Let me wait for some suggestions. I'll be happy to receive some answer. What do you think, or how do you think this feature is important before I say it? Yeah, thanks for the responses. They were really good. And the importance of this. So you can see that our amino acid has two ends, and the two ends respond differently basing on the behavior, the medium at that particular point. Whereby the way it responds when it is in acidic medium and the way it responds when it is in basic medium is totally different. Okay? Totally different. Not so? Yes. So it has that ability to exist in this state. So that point where you are equal, then we call it the isoelectric point of an amino acid, and it is good in buffer systems. In that, for instance, let's say our amino acid, which is like this, you have the NH3, okay, and then the COO. Oh, you have lost this. So you have the COO. So this solution or this compound, this amino acid, it is going to act as a buffer, or it is going to buffer a given solution to resist changes in pH when a little acid or a little base is added. In that, when a little base is added, for instance, okay, and you know a base has hydrogen ions, okay, so the hydroxyl ions, this is a base. Now the OH ions will react with the hydrogen ions that were lost from this, okay, it will react with hydrogen ions that were lost from this carboxylic acid to form water. And water is a neutral compound, it's a neutral molecule compound, which won't have any effect on the overall pH of that particular cell. And so as a result, you have buffered. A buffer solution is that solution that resists changes in pH when a little acid and a little base is added. So here you've added a little base in this solution, and a little base will react with the free hydrogen ions in this solution to form water. And water is a neutral molecule, so won't impact on the pH. Okay? Similarly, when a little acid is added, okay, now the little acid will react with the base, the amino group, okay, it will receive that lone pair of electrons from the amino group to form what we call this with a plus. And so you have removed the hydrogen ions from the solution. Now take an example where somebody is involved in a strenuous exercise. Okay? Exercise. Whereby in a strenuous exercise, carbon dioxide is going to be produced, enough of it, and the carbon dioxide will react with the water. Sorry. The carbon dioxide will react the water to form the weak carbonic acid. Okay? Which partially dissociates to give us the hydrogen ions and the hydrogen carbonate ions. Now, do you see these hydrogen ions that are produced from carbon dioxide that is produced in the tissues? You can see that. Now, these hydrogen ions, they can alter the cell, the pH of the cell. Remember, pH is measured as negative log of concentration of hydrogen ions. Okay? But when you remove these hydrogen ions from the solution by combining them with this amino group and you form this compound, then this compound won't have any change in the pH. And so it will be buffered. Similarly, towards the lungs, when you break down the hydrogen carbonate ions to carbonate ions or to carbon dioxide, you do this reversal. So it leads to the formation of many OH ions. Okay? And these OH ions might alter the pH. So the free OH ions, they will combine with the hydrogen ions, okay, the free hydrogen ions within the aqueous solution to form water, which is a neutral molecule. Hence, you have buffered the temperature. There won't be any rise or any decrease in the temperature, in the pH, sorry, which wouldn't favor our protein. Is that okay? Because imagine if you alter the pH of blood. Remember, pH, when you change pH, the solubility changes. You have there proteins, other proteins. The proteins get denatured. They get deactivated. So, in other words, the internal temperature. What's wrong with temperature? The internal environment. And that environment of blood must be kept constant. And how do you keep that? Same reaction. Buffer system. You know that in the blood you have there hemoglobin. Now it is hemoglobin that has most of these groups exposed, okay, to easily carry out that buffering system. Any query for me? Anything that I should talk about? So this concept is really good. This concept of buffer is really good. It's really good. So let's talk about the types. And like I told you, there are 20 known amino acids. And of the 20, we have 10 essential and 10 non-essential. And of the essential now, you don't need to cram all these amino acids. Okay? Nobody's going to. Maybe when you are studying biochemistry for the if you are doing biochemistry, then, but for A-level biology, no, no, no. You don't need to cram all these 20 amino acids. Okay? So now these are the essential amino acids. Like the same way we defined essential fatty acids. These ones, your body cannot make them. Okay? So by all means, you have to obtain them from the diet that you're going to eat. In that if you eat poorly, then you won't have these amino acids. And so you become malnourished. Because once you are forming a given protein, and you are supposed to put proline or varanine or lysine, and you don't have the lysine in your body, and your body cannot make the lysine, then that protein stops. If you are making maybe any protein, and you are supposed to put proline or phenyl alanine, not phenyl amine, and you don't have the phenyl alanine in your body, and you cannot make the phenyl alanine, then that protein stops. You cannot move further. You cannot proceed further with it. So therefore, these essential amino acids must be obtained in the diet. So make sure you guys have some balanced diet. If you eat meat today, tomorrow eat this. Next time eat, be a vegetarian. Because most of these ones are found in vegetables. Next time be a vegetarian. The other time be this. Try to eat various foods. So that you try to bring these amino acids into your body. Then the non-essential ones, those ones which we can make in our bodies, we have tyrosine. Now, like I told you, no one is going to ask you to write these. Okay? No one is going to say write the structure of glycine. Even me, I don't know the structure. Write the structure of cysteine, aspartic acid, glutamic acid. No, nobody is going to tell you write this one. Okay? As long as you know the essential and non-essential amino acids, then you'll be good to go. Let's form a polypeptide chain and then we call it a day. Now, from what we've been talking about as far as carbohydrates, as far as fats are concerned, it is the same condensation reaction, and the reaction needs formation of a water molecule. So can you try to show how these amino acids are going to condense together to form a polypeptide chain? Try before you condense. Try. I'll give you just something like 2 minutes to do that. Just use 2 amino acids. Okay? Yeah, so I've seen your work and it was fair. It was fair. Okay? Yes. Now, you people, condensation occurs between the carboxyl group and the amino group. It's between the carboxyl group and the amino group. And boom! I saw some funny stuff. It's okay.
Maybe we try again.
You want to try again? It's okay. Let's do. Sorry. So condensation occurs between the amino group and the carboxyl group. One of the hydrogens on the amino group condenses with the the OH group on the carboxyl group, and what is formed is a peptide bond. And I saw funny stuff and I was almost bursting, but it's okay, I cannot burst into rafters because we are learning, and we start from scratch, not from scratch. We start with what is known to what is unknown. And after that condensation, it's a peptide bond that is formed. Or if there are two, then you can be specific and say a dipeptide bond. Now, further condensation. If these amino acids, maybe if you have, I'm going to illustrate. If you have amino acid A and amino acid A condenses with amino acid B, then you're going to form a dipeptide bond or a peptide bond, okay, between the two amino acids. But if you have further condensation and the amino acid B condenses with C, C condenses with D, D condenses with MAB, A condenses with C maybe, there is no order, okay? Maybe here you add Z, maybe here you add F, maybe here you add H. So further condensation of these amino acids, then we shall form a polypeptide chain, okay? Polypeptide chain, okay? There is no E. And the bond is the peptide bond. So it is as simple as, I think this is the easiest condensation that you can ever encounter as far as condensation is concerned. So you can have a look. This is your amino acid A, okay? And this is its structure. You see we have an amino group and then the carboxyl group, okay? So the amino group has condensed with a carboxyl group. You can see to form a peptide bond. Maybe before this, let's have a look at this. So the one of the H on the amino group, it will condense with the OH, okay? Whereby it will go away and form water molecule. So the bond that is formed, it's a peptide bond, okay? It's a peptide bond. So the same thing happened here. So you can further condense, same thing, okay? And you form a polypeptide chain, okay? Which is a chain of very many amino acids. So now this is the R group, okay? And the R group is in this amino acid, is like this. And this is another R group. And the R group in this amino acid, it's like this. And this is the R group. And the R group in this amino acid, it's like this. So all amino acids have an amino group and a carboxyl group. However, the R, it is the one that varies from amino acid A to amino acid B to amino acid C. Okay? So if they are not, so somebody was asking that when we remove this water molecule, the carbon bonds directly to nitrogen. And I was like, yes, the carbon bonds to nitrogen, and the bond that is formed is a peptide bond. Okay? It's a peptide bond. And it's the one that holds these protein molecules together. Okay? In a polypeptide chain. And so there is some important points to know. There are three of them. Number one, the R group orientation determines which protein is going to be formed and how complex that protein is formed. We are going to see. We are going to see what this point means in our proceeding videos, in our proceeding lessons. Okay? Number two, when you are reading the directionality of a given amino acid, we have the N terminal and the carbon terminal. Because see, it starts with the N and you have the amino group here, and then condenses. It must end with the carboxyl group. So therefore this side, it is the N terminus, and this side, it is the C terminus. So therefore you can identify these amino acids basing on these ends. Okay? Then finally, the flexibility, I think, of a given polypeptide chain. In that the peptide bond that is going to be formed, because we have said that it is formed between the carbon and the nitrogen. Okay? So this is a peptide bond and the rest are normal covalent bonds. So this peptide bond that is formed, it is highly rigid. Okay? But these adjacent covalent bonds, they can flex a bit. Okay? They are flexible. And you know they are the ones that are many. Hence, they make the overall structure of a protein to be flexible. Okay? Yes. So consider this polypeptide chain, for instance, where this is the amino group, this is the N terminus, and this is the carboxyl group, the C terminus. Okay? Where one C has lost its hydrogen and it has attached on the amino group this side. So now this is the C, this is the hydrogen, this is the R group. So this is our peptide bond. This is our peptide bond. Our peptide bond. The same. So these peptide bonds, they are rigid. Okay? They are rigid. But now these covalent bonds that are adjacent, these neighboring covalent bonds, these ones, they are flexible, and so the whole protein structure therefore is going to be flexible. Okay? So it can easily be molded, it can be easily coiled. We shall see the proteins and their structures. So basically this is where we are going to stop from. And yeah, we can meet in our
Excellent.
next lesson. Yes.